record. Okay, so apart from planning of experiment here, number 1 until number 7, where you conduct experiment, after you conduct experiment, the second section is you'll be asked to present your results from the experiment. So you need to be able to draw table or graph, okay, and you need to use the correct format to fill in your table and graph. Cannot simply do one, okay? There is a format to follow. Where to put your independent variable, where to put your dependent variable, okay? Followed by the unit must be stated there. Then describe and explain the results in your table and graph. So this is from the theory. So from the theory in AS that you have learned, usually what they were, the topic of the experiment that they ask you to do usually will be around enzymes, biochemical tests, diffusion and osmosis. So for enzymes, usually they ask you to test effect of substrate concentration or enzyme conjugation or pH or inhibitor or temperature on the rate of reaction. Okay. Biochemical tests, you know that these are the basic biochemical tests that you need to be able to conduct: reducing sugars, non-reducing sugar, lipids, proteins, and one more is actually the starch. Okay, the starch. Okay, then we also got experiment that asks you to conduct experiment for diffusion and osmosis. So they can give you whisking tubings, potato cubes or discs. Okay, because when potato and disc, when water enters, it will become bigger. Okay, the length will increase, for example. So you need to measure the length after a certain period of time. Okay, onion epidermal cell. So you can look under the microscope, put it under the microscope, and you see that epidermal cells becomes plasmolized or becomes turgid. How many percentage of cells are plasmolized? Okay. Yeah. So I do not know whether you know about this graph or not. When you look at the percentage of cell plasmolized, it will tell you what is the water potential of the cell. So this is the concentration of sucrose solution. Okay, probably in molar. And then this is the percentage of cell that's plasmolized. Okay, so when you plot the graph, you expect to see that the graph will look like this. I think if it's correct. So if you are asked, so which is the sucrose solution that has got the same water potential as the cytoplasm of the cell? Which is the sucrose solution which has got the same water potential as the cell? So which one do you think is the answer? Jung Seong Myun.
What are the options again?
The option is on the graph, so you have to tell me.
This is a good question.
Huh?
I'm not sure.
Give a guess, ma.
Oh, can I get the question again?
How do you find, okay, which sucrose solution will give you, which concentration of sucrose will have the same water potential as the cell cytoplasm? Okay, Joey. Give me all the 3 possible outcomes when a plant cell is immersed in sucrose solution of different concentration. 3 possible. Give me.
Plasmolysis.
Okay, so it can be plasmolysed. Some more.
Turgid.
Turgid. Some more.
And nothing normal in its concept.
So incipient plasmolysis, which is equals to normal. Okay, so this is theory, right? You learned already. A plant cell immersed in a sucrose solution, it can be plasmolysed because it is high concentration of sucrose. How high is high? Yep, 支气.
Yes.
How high is high?
What is high?
What is not paying attention? Myra?
Yes. Miss, can you repeat the question? What the concentration of sucrose?
Yes, that's right.
Like above.
Of what?
If it's higher than the concentration of the, I also don't know what means.
Sucrose concentration of is higher than what? When will the cell be plasmolysed when it's immersed in a sucrose solution?
When the water potential is low, like the concentration of sucrose is more.
More. If you say you want to compare with something, you need to give the full statement. What are you comparing with?
More than
what?
50% of the sucrose. I mean, like maybe it's the sucrose concentration in the in the solution is higher than the sucrose concentration in the cell.
Okay, sucrose concentration in the surrounding is higher than concentration of solutes inside the cell, because cell is not only made of sucrose, it's all the solutes in the cell that will contribute to water potential of the cell. So therefore, if the cell is plasmolysed, that means psi of the surrounding, which is environment, is actually less than psi of the cell. So that's why there's a net movement of water out of the cell, so the cell is plasmolysed. This is very basic. Okay, so that means the sucrose concentration is higher than the solute concentration of the cell. So that's why the cell is plasmolysed, right? So what if case number 2, where you say that the cell is normal, it's not plasmolysed, it is not turgid, it is just normal. So what is the sucrose concentration versus solute concentration of the cell? Najiha.
It's the same.
It's the same, right? So it's the same. So therefore, it's the same. Then what about when you said number 3, the cell can be turgid? So when the cell can be turgid, what is the difference between sucrose concentration and solute concentration in the cell? Lô Vân Thảo.
When it's turgid, the water, the solute concentration inside the cell is lesser than the sucrose concentration surrounding.
Yeah, correct. So you're talking about concentration is, hang on. Solute concentration is less, is it?
Yes, that's why sucrose concentrate. Right. Where outside is less, inside is more. I think the other way around.
Yeah, correct. A cell can be turgid because the cell is placed in a solution where psi of the surrounding is higher than psi of the cell. Okay, so psi of environment higher than cell, so there's a net movement of water into the cell. So therefore the cell will swell and becomes turgid for plant cell. So it means the original sucrose concentration, the original solute concentration inside the cell, is it higher or lower than the environment? Dafina?
The solute of the cell is higher than the sucrose
Correct.
concentration.
The original solute concentration in the cell is higher than the surrounding. That's why there's a net water movement into the cell. So these are the basic knowledge that if they ask you about osmosis and diffusion, you need to be able to use all the keywords to describe. Okay, which one is higher than which one? So there's net movement of water from where to where through the cell surface membrane by osmosis. So these are the keywords. From where to where, through where, by what process. So here, I already given you there are 3 possible cases of the cell immersed in solution of different concentration. 1, 2, 3. So tell me, which is the location where you can find out that the sucrose concentration is the same as the concentration of solute inside the cell. In this graph, where the y-axis is percentage cells that are plasmolized. And this is 100% here. This is 0 here. This is 100. So where do you look for the region where the concentration of sucrose is the same as the concentration of solute? It's very easy, yeah. Okay, Abner, go. Abner, go. Okay, no response. Crystal Liu?
Yeah, middle
Where?
part.
Middle part. So middle part is how many percent?
How many percent is same as the solvent, like the cell and the surrounding is the same. That is 50%.
50% of what?
Of the sucrose concentration. I don't know.
There's no 50% sucrose concentration here. My sucrose is in molar.
Solvent. I mean, like the water.
There's no water here indication that 50% water. I can't find this from the graph. I want to find from the graph.
50% cells plasmolized.
Correct.
Bro-
50% cell plasmolized. It's very obvious, isn't it? Right in the middle, because I already showed you there are only 3 possible cases. Either the cell is plasmolized or normal or turgid. So among all these 100% must be right in the center is the one that is normal, isn't it? So the one normal is 50%. So in the slide, when you count 10 cells, out of 10 cells, when 5 cells are plasmolized, it is that solution that has the same water potential as the cytoplasm of the cell. So this is another possible experiment that they can give you. Okay. So when 50% of the cell is plasmolized, then the solution is considered as having same water potential of the cell. So therefore, you interpolate the graph, look for the concentration of the sucrose solution here, and then there is a table of conversion. A table of conversion means there's a table with the concentration of the sucrose on the left side in molar. Okay. And then on the right side, it is a water potential psi. So it's a water potential here in kilopascal, stroke kilopascal. You need to know how to write the heading of a table correctly. Always the title stroke followed by the unit. Title stroke followed by the unit. Anything missing, no mark. Okay. So from here, if you look at sucrose, okay, sucrose is this value. Go to this table to check for the value of the sucrose. And then from there, read the water potential psi. So that will give you the water potential of the cytoplasm of the cell. Okay. So that is for osmosis. So let's move on from here. So you will have result that you need to present, you need to describe, you need to explain your results. Why does it go up? Why does it go down using your theory? Okay. effect of temperature, effect of pH. Then identify possible sources of error and how to improve it. So like what I say here, if the question asks you to maintain temperature between 30 and 40 degrees Celsius, this is definitely a source of error, because 30 degrees Celsius when you conduct experiment versus 40 degrees Celsius is totally different rate of reaction already. So when you maintain your temperature at a range, you are introducing an error, because different temperature, different kinetic energy, different rate of reaction. So the question deliberately gives you instruction to conduct the experiment within this temperature, and after that they ask you to identify error. So sometimes they will ask you to do it like this. Okay, or for example, you're supposed to find the endpoint where the pH changes from acidic to becomes neutral. So they give you an agar block, which has got naphthalene, okay, and then it is like pink color or blue color, depending on whether acidic or basic. Then you're supposed to put in a solution of acid or base, and then when it diffuses in, the color will change until it becomes neutral. So sometimes the change of color, especially the end part, where the final part of the color disappears, it's very difficult to determine exactly what is the time that the color totally disappears, because it's already very, very light. It turns from pink to become lighter and lighter and lighter and lighter. So you do not know exactly what is the time for the end point where the last bit of the pink color disappear. And this always will be the significant source of error when you need to determine the timing when a color disappear or color change. That one is very difficult to determine one. So that's always the significant source of error. Okay. So this is example only, example. How to improve experiment. Okay, finally, they will ask you to do a conclusion. So after that, conclusion, so they can ask you to do conclusion. That means based on the results, based on the hypothesis, as well as the result that you obtain, based on these two, please do the conclusion. So does the result support the hypothesis? Does the result negate the hypothesis? That means, does the result support or partially support, or where's my or negate the hypothesis? Negate means does not support. Okay. So if you say, okay, as temperature increases, the rate of reaction increases. So I'm expecting my result to be as temperature, which is my independent variable, must always be placed here. And this is a dependent variable, say the rate of reaction, which you count the total number of bubbles, okay? Number of bubbles per minute. So your unit is per minute -1, okay? So if I say my hypothesis is as temperature increases, the number of bubble released per minute will increase. So I'm expecting my result to be like this. But when I conduct experiment, I obtain a result which is not like this, but my result is like this. It goes up this way, and then it comes down very quickly. So therefore, when you want to do conclusion, you need to conclude based on your result versus the hypothesis. So your result shows that, okay, for my temperature, which is within here, my result supports the hypothesis, whereby as I increase the temperature, my number of bubble released per minute also increases. I don't care whether it's increasing this way or this way or this way, they are all increasing. So it supports my hypothesis. So you say that, okay, on the conclusion, you say that the result partially support the hypothesis. And then you say that from which temperature T1 to which temperature T2, the result actually support the hypothesis. And then from which T2 temperature to the final temperature T3, the result does not support the hypothesis. It shows that within this temperature, as temperature increases, the number of bubbles released per minute actually decrease. So this is how you need to do your conclusion based on the result. So say whether your results support, partially support, or totally negate the hypothesis, and then use the result there to support your conclusion. If you say support, tell me where does it support. So this part here support. If you say partially support, which part support, which part does not support. If you say negate, tell me, so what is the result that you get that is opposite from your hypothesis? So that means you have to only give, that means your result is like this. So it's totally opposite. So that is making conclusion. Okay, so you can be asked to make conclusion. And then one more thing they can ask you to do is that they say that if time permits, if there's still extra time, they want you to test. So how do you conduct experiment to test effect of pH on the rate of reaction? So they can ask you to write out how you're going to conduct experiment to test for a different variable as the independent variable. So this is another thing that they can ask you to do. Why? Because in paper 3, they will give you this instruction. All these instructions will be given to you, except for biochemical tests where they expect you to know how to do. They may even ask you to write the procedure. Other than that, they will give you the procedure. So because they've given you the procedure, they may give you an additional question which you need to write the very basic procedure. Okay, the very basic procedure. How do you test for effect of pH? So you talk about what is your independent variable? What are the 5 values you're going to use? You need to say the 5 values for the pH. And then, so what is the temperature? What is it you want to measure? You know you want to measure number of bubbles, so you have to write down. Then what about your standardized variables? So what are the variables you want to standardize? If you say temperature, so what is the temperature you're going to use? Of course, you're going to use optimum temperature. So based on your first experiment, you know what is the optimum temperature. You use your optimum temperature because the rate of reaction is the fastest. You want to get your result with the minimum time, minimum money, minimum effort, and maximum result. So you need to choose the optimum temperature if you know. Okay, so what are your standardized variable here? What are things you need to standardize? Okay, so these are the full component of your practical paper, which is paper 3. Full component is inside here. I've mentioned everything. Okay, right. So therefore, you have to practice so that you become better. It's all about this only, nothing else, because this is about practical. Okay, so is this I have to save so you can get it, isn't it? I better save. Okay, save this already. Okay, so now today we are going to continue to look at the questions.